So Dr. Mazzeo, okay, you've been studying exosomes for 22 plus years.
Yes.
What drew you into studying exosomes at 22 years ago?
Yes, starting from the beginning, what brings me to exosomes was my project for my PhD studies. That was related with the biogenesis of exosomes. And back to 2004, 3. And at that time, exosomes were very controversial. Yes, also in between the in the academia, in the scientific field. So for me, it was very challenging. Yes, to start my thesis with this subject,
Yeah.
and also with the difficulty that maybe because scientists were telling me, no, you are just study the breezes. This is nothing. They don't do nothing. And I say, well, I have here the data. I have here results that I'm obtaining for my research. So I was really and truly believe on my results and what I was seeing at that point. But it was very, very, very difficult to continue that until, MISEV appeared, which is the International Society for Extracellular Vesicles. And at that point is when, all the exosome extracellular vesicles become strong. But when I was studying them, and during my thesis, 4 years of study, the risk was that suddenly, yes, the scientific field decide, okay, this is not something that could be financial debt to get together the finance for that and to obtain grants, and maybe the grants for that study will cut and also my study. So it was very, very challenging, but I totally believe on this science, believe on my results and my colleagues' results. So together we started to create this different kind of communities to share exactly what we were discovering at that time, and later the creation of the International Society for Extracellular Vesicles that I'm part since that was created, because I'm also doing research before that was created, and after that I published with them, and I also publication in the journal that they have now. But together we put the reality and the discovering, yes, on the table to defend the science. And yes, I wanted to add other thing that come to my mind. That also during that period, the difficulty was in relation of the technology. As I always say, we have to go with the technology. Things will progress, but we have to be patient. So at that time, we were not using nanotrichine analysis. We were not the characterization of the exosomes. And I say at that point was not a guidance to characterize the exosome or how to do the things. We had to put all the things in order. Yeah, so it was very difficult at that point because we were doing the research and we wanted to show, yes, that the exosomes were there. The main characterization at that point was based more on transmission electron microscopy, because and I have picture of that beautiful picture from the cell, and then later it's a pity that my skin that I was preparing on my computer I can, but I hope later we can see that. How from the electron microscopy you can also see how the multivesicular bodies that will secrete the exosomes fusion the cell and secrete the exosomes, and you can see the exosome and the structure of the exosome, the morphology. That was fundamental. So that is how, and well, we managed to do and to progress doing research until nowadays that's they start to be a trend topic now.
Yeah.
From that, in relation of my career in this, I was studied them in different laboratories around the world, fascinated with this field, and also in different. I will tell later in relation of the exosomes, all the potentialities that they have, and I was studying this in all those potentialities, like diagnostics, therapeutics, and medical as a medical device.
Yeah.
So yes, passionate about that, all those years, and still.
That's awesome. I mean, you have to love what you do, right?
Yeah.
I'm a strong believer in
Yes.
that, because if you're
I am.
passionate about it, you wake up every day wanting to do more and more and continue doing that, right? One thing that you mentioned was that you said that, I mean, I believe, in my opinion, is that exosomes are controversial till this day. You mentioned that they were controversial before as well when you first started. What has changed? Like they're controversial today. What was the difference between the controversial when you started to today?
Well, now the controversial is totally different. Now we have proof, we have science just in behind, because all the hard work during all those years, 30 years of a specific study of the exosome. But now the controversial part is not right now is not something controversial that the exosomes that the cell secrete exosomes, they are not trash, and they do one specific thing outside the cell. So now the controversial part is in relation of how they are commercialized, how they are right now. In reality, is not that. It's how we pass from the science, for the knowledge that we have in the science, to the industry, and to go to that process is still is the thing that had to improve. I have to as I say before, the technology also have to improve, and that is the part that is controversial in relation of, okay, this is the science, and now how we are going to scale up. Yes, the exosomes to use, for example, in therapeutics. How we are going to purify the exosome in a high scale. How we are going to standardize this. How we are going to in relation of conserve, yes, the exome, the conservation of the exosome, in relation of the reproducibility, all the things is the part that is controversial, because in all those steps, what's happened is that on the way, for example, to scale up, or or for example to in relation to the conservation, that is against the potency of the exosomes. In more those steps, we call the potency of the exosomes could be reduced. So that is the challenge right now, and the controversial part, how we are going to do that. And also, well, that is from one part, but also is how nowadays are used are using. Yes, most of the controversial in that part is misinformation about the exosome in general and the extracellular vesicles. The difference between the extracellular vesicles that exosomes are part. Yes, exosomes are an extracellular vesicle, but the difference between the others. And how they are used in therapeutics and aesthetics, how they are used.
Okay. Okay. So I would like to start kind of with the basics, right? So we hear, yes, we hear the word exosome, we hear the word extracellular vesicles, ESEV, MISEV, they've recommended to abandon the word exosomes in certain terminology unless we know how to classify them, right? Unless we know it's for certain, right?
Yeah.
Um, what is this? I believe there's going to be two questions. So, what is an extracellular vesicle, and then what is an exosome, right? In because we know from what I've read, an exosome is an exosome is an extracellular vesicle that is measured between 50 nanometers to 150, and then they have certain functions. So, what is your definition? And then, how are they different from other extracellular vesicles?
Yes. Yes, it's a pity because I have a beautiful scheme. My computer, want to start now, so maybe we have lucky and we can pass that, but I will explain without that
The
figure.
diagram.
Yes. So my definition for the exosome is that exosomes are the functional part of the extracellular vesicles. That is my definition. Most of the definitions are based on the characterization of the exosome. They say, well, they are vesicles that the cell secrete, and they have a specific cargo that the cell want to transmit. So exosomes are also, well, you say the functional part, function. What is the function? Cell-to-cell communication, yes? So, and how they do that is through two main functions that they have. One is cargo delivery, and the other is reprogramming that receptor cell. So going to the start of the basic, the biology, the exosomes, and also, as you asked me, the difference between other extracellular vesicles. The main difference, to know the main difference, we need to back to the basic, to the biology, which is inside cell biology, which is what's happening inside the cell. And that I will explain as much basic as I can. And is needed to know because the exosomes are the only extracellular vesicles that are made inside the cell for one specific purpose. For that reason, I repeat my definition. Extracellular exosomes are the functional part of extracellular vesicles. The rest of extracellular vesicles, most of them come from the membrane of the cell. But specifically, the exosomes come from one pathway, and it's a different compartment. It's like if you imagine the cell as an apartment, the different compartment, the different rooms that you have in your apartment. Yes. So they create this kind of different compartments inside the cells that are called endosomes inside the cell. So this is called endosome pathway. So how the exosomes are made is with a endosome pathway, where these compartments are made, and inside of this compartment is where the vesicles are created. All this inside the cell, yes, inside. And when they are created inside the cell, the exosomes are called intraluminal vesicles, yes? And it's inside those compartments. This specific compartment that has the intraluminal vesicles that will be secreted as exosomes is called multivesicular body. Yeah? When this multivesicular body is created inside the cell by invagination of the membrane, well, it's a process, it's better with the skin, so I'm not going to be more to the point. But the main thing, what I want to say at this point is that in this process is when the cell decide which cargo will put inside the exosomes to deliver to other cells. Yes? Is inside the cell in this specific process. There is a specific pathway, a specific process that the cell do for one specific function, which is delivery and cell-to-cell communication. Yes, and communicate with other cells. That is the main difference. Is all this process to create these vesicles that will mix with the other extracellular vesicles that come from the membrane of the cell that have other more basic function, but it's not this cell-to-cell communication. They help, altogether help, but the main are the exosome because are created inside the cell for that specific function. That is the main
Got it.
description of that.
Interesting. Yeah. So the function, reprogramming and delivery are the two main function.
Exactly.
Okay.
So, well, they communicate. Main function in general, as everybody knows and say, is cell-to-cell communication. How they communicate is delivering cargo to that receptor cell, and one delivery also starts signaling it inside the cell and reprogramming, have the capacity reprogramming the cell, the receptors.
Okay. Got it.
That is how they communicate and what they do.
Yep. That's what, okay. How are exosomes characterized and identified in a lab setting?
Yes, good question. Well, first I have to say that the characterization is part of the MISEV guidelines. So after all these years, we were improving this whole, as I say from the start, from my studies. Well, we need to characterize, we need to show that this is an exosome, how we do that. So during all this time, all these years, we were improving the way that we can characterize. And also, like yes, the nanotechnology was improving with us. So altogether, we are now with the technology that we have that will improve, and how we characterize right now is fundamental. And here I want to really talk about something serious. Well, all this is serious, but
This is like very
this
serious.
is main point now.
Yeah.
This is main point because
Yeah.
not everybody understands.
Yeah,
this.
because I feel like when they're characterized properly, then that goes from whoever's purchasing and buying, and then from the patient receiving the product. So you want to make sure that whatever
No,
product you're
exactly. That
selling,
you
right?
have the characterization is to say that you have exosomes in your sample. That is how you have to show that you have that. As a scientist, we need when we write our manuscript to present in a journal or for a study, and if all our study was made based on exosome, we need to characterize. We need to show that we characterize the exosomes, and we were working with exosome, and our results were because of the exosomes. So if we don't do that, our manuscript is rejected, and we can put exosomes in the title.
Oh, but then you have companies.
If
But then,
the characterization is not there, it's
yeah,
rejected. It's
interesting.
rejected.
So that which I think is good, right? Because that's setting good standards, right? But then that standard should be applied to the entire industry, to the companies producing it,
Exactly.
and yeah.
That is the
So
thing. Because now yes, I found lot of companies that put exosomes, and I say, okay, and the characterization, and they say, no. Well, we will talk about that.
okay,
We have time to talk about that.
yeah, okay.
But that is the main point.
Question.
So the characterizations. Yes, we go to the main point. How we characterize the exosomes. As I say, that this come from all those years of studies of improvement and also with the technology. So now we have on our technology we use, it's not new, but for extracellular vesicles are for this specifically the nanotrichine analysis. It's a really great nanotechnology improvement that permits us. Before, when I started, that was not accessible. So now we have this equipment that is based on nanotracking analysis that give us the possibility to differentiate the different vesicles in relation of the size. Yes. So we can characterize and differentiate the exosome by the size. The other technique that must be used is the transmission electron microscopy that also differentiate the exosomes from other vesicles in relation of the structure, the morphology, yes, how the double membrane, how they looks like, and also the markers that come from the biogenesis. And I will also later to explain the difference when that comes from the biogenesis or and other cargoes that they transport. So those are the main and well how we most of the cargo, the markers are from proteomics, different techniques on proteomics. So those are the three techniques: nanotracking analysis, the transmission electron microscopy, and the proteomics. Why? Because the exosomes have this characteristic. All exosomes in structure and function are this. And function, we will talk about that later also. But they are the same in relation of how they were made, in relation of the biogenesis, are all the same. So they have one specific size and a specific morphology, and they transport a specific markers that come, specific from the biogenesis. So we can, with those three techniques, we can differentiate for other cells. It's not just the size, because there are other extracellular vesicles that have the same size of exosomes. So just the size is not telling us nothing. Just the morphology, no, because again, other things, you have also to have an expert eye to really differentiate vesicles of the same size and the electron transmission electron microscopy, and differentiate just from one image to other vesicles. And the other is the markers, but there are also some other extracellular vesicles that have those markers. So you really need to do the three of them and the three of them have to be checked, yes, to say this is an exosome.
Okay.
There's not just one or two, it's the three of them.
So you said the size, but the size, the transmission electron microscopy, that's what that one, and then the markers, right?
Yes. And also through, I love to do through the electron microscopy, you can also do the marker, see the marker, because there is a technique in the electron microscopy that you can, label the marker, the biogenesis marker, and you can see on the image that they are the morphology with the marker.
Okay. Interesting. So essentially, if in the way that I'm interpreting what you just mentioned is that if a company is categorizing their exosomes, right, or their extracellular vesicles by size only, that might not be the best option, correct?
Yes, there is more to talk about that. I don't know now or later.
We can, yes, and then the transmission. Okay, so now we were talking off camera, and I know that said what I've read is that many, almost every cell in the body, and you said most cells in the body secrete exosomes, right? So does the source matter where that exosome is coming from?
Good. Yes, totally. Because as you say, exosomes are secreted for most of the cells of our body.
Yeah.
And that means that the information, as I say, they are cell-to-cell communication. So the information that they are going to deliver is in relation of the cell, yes, from where they come. Yeah, you can, if you have an apple tree, the apple tree will give you apples. If you have a pear tree, will give you pear. Yes, you can. Yes, so it's what the cell contain and what the cell can transmit to other cells.
Interesting.
So there are. The source is important. Again, as I say, the source is important because that will depend of what they are going to deliver as a cargo and which information will be delivered in relation to reprogram in some way the acceptor cell. So it's totally different. The exosomes in relation of it, the exosome is the same. I have to make a point here. There's the morphology, the biogenesis markers, the size, the same. But the cargo and the kind of reprogramming is totally different depending of the source. Yeah, it's clear.
Yep, that's clear. Okay, beautiful. Thank you for that. So then if we are using, let's say, autologous products, then those exosomes are very crucial because they're coming usually either adipose or bone marrow or blood.
Well,
And then
yeah.
those matter. And then I actually saw that our urine also secretes exosomes or has exosomes. Yeah, that's insane.
Exosomes. You can have exosomes in your urine. You can have exosome in your saliva. You can have exosome in your semen. You can have exosome, in the blood, of course. You can have exosomes. I say in your saliva. In most of your fluids, you can find the exosome, and also later also we will talk about that. Also, the cancerous cells secrete exosomes a lot. Yes, for that reason, and also cells that come from other illness. Yes, for that reason, the exosomes can be used and are using now. They are kits used as a diagnostic. Yes, early. They are early biomarkers. That is, I want to differentiate the function of the exosomes, which is they can deliver cargo and also reprogram the cell. But the uses of the potency that the exosome has in medicine is they can be used as a diagnostics, therapeutics, and as a drug delivery.
Wow,
Yes.
that's awesome.
For that, yeah. And that is because of that, because most of the cells secrete them, so we can use them in different ways. Why they are important in diagnostic? Also, again, because of the cargo. Putting an example, the cancer cell. The cancer cell, they deliver exosomes with markers, yes, for that specific cancer cell, yes, for that specific type of cancer. So from that, we can purify the exosomes and check if the marker of that cancer cell is there. Why? Well, now you can do that, yes, for the markers that are freely in the blood. Why exosomes? And I say because exosomes, when just the cancer start, the cancerous cells start to divide, the cancer cells start to secrete the exosomes, and in a bunch. So you can very early detect that on exosome. If you purify the exosome, concentrate for that marker to be detectable, because the marker had to be just detectable in the sample that you took, in the blood sample, for example. You purify the exosomes, and you can detect earlier. There is now a kit for a wonderful company that create those kits for prostate cancer right now.
Interesting.
Based on exosomes
For diagnostic purposes.
in urine, as you say. The exosome from the urine I use right now. There is a kit that is used for early detection of prostate cancer.
Wow, I have not heard of that. Thanks for sharing that.
Yeah.
That's cool. That's cool. Okay. There's so much misinformation out there about exosomes, extracellular vesicles, and what are the biggest myths or misconceptions that you see out there?
Oh, yeah. Wait, because I did a long list. I don't want to miss any.
You don't want to miss all of them. There's so many of them. Also, too, like if you guys, everyone that is listening, if you guys can probably drop something in the comments to say
Yeah. Oh,
what are
yes,
some
please.
misconceptions, please. I want to hear what you
Yes.
guys have seen, any
I
like
like
that. Yeah, I
me too.
want to keep this very engaging, so we can all learn, right? So I
Exactly.
appreciate it.
Um, Well, one is starting from what we were talking in relation of the cargo, that whatever cell that you start to produce the exosomes, they will have as a cargo the same thing as whatever other cell. So the source doesn't matter. Wrong. Yeah.
Okay.
That is one important thing. So the source, yes, matters a lot. Everything depends of that. And I want to make a point here before I forget is that also the same source can give you different exosomes in relation of cargo. That depends of the environment that you or how you grow those cells. If that change, the cargo that they deliver, the information that deliver, will change also. Yes,
Interesting.
because if it's like I'm
It's
gonna say
like, for example, I mean, the way that in basic terms, the how I interpret that is that if we are outside and let's say something scares us, our flight or flight turns on, what happens to our heart rate? It goes up, right? And
sorry.
so our internal, all that kind of changes, right? So similarly, you can that
Well,
same concept can be applied.
yes, you can do that in relation of communication. Is what the exosomes do is that if you have a happy environment and a
Environment.
happy day, you are going to start to speak, yes, and to communicate very happily.
Yeah.
But if you are in a bad
Bad day.
environment and you
Yeah.
are not good and know what you are going to speak, not so good, or at least no, yeah.
Yeah. That's true. Yeah.
So the same with the cell.
Yeah.
So depend that, and that is in relation of all of the one of the problems that the company have right now. How that to maintain all the standards in the production, to maintain the cells in exactly the same growing in exactly the same in exactly with the same conditions, yes, for those exosomes to be, and they are doing some changes. And as I say, sometime to maintain that, they goes against or that changes start to affect the potency of those exosomes. We are going to to talk later because there are some specific point that I later to punctualize. So yes, what the other misconception is in relation of the number. Yes, the quantity that you have in the vial, and they say yes, we have 2,000 billion of exosomes, and they say, well, in reality, I hope that you have that, but right now I have to back that from the start. It's like how we and the scientists and also from the industry have that number is using the nanotracking analysis. Yes, one part of the characterization. But that analysis, what tell us is that you have particles per milliliter. Particles. Yes. So And this machine can separate. Yes, you have to do other
It
things.
can
You can.
or it cannot separate?
They cannot
It
basically.
cannot.
Yes,
Okay.
basically, you can do an extra study, and it's more an indirect detection of that content. Yes, labeling the exosomes and that, and you can have an idea of in all that um, um,
What's in it?
measurement that you are doing with that sample, how much exosomes you have. But directly, you can't. You can't. Most of the information that we have is we have particles, will be um, extracellular vesicles. You say extracellular vesicles if you did something before, which is the purification. Yes, we didn't talk yet about that, but
Okay.
not all the companies do that. We in science, they if they're researching about exosomes, the exosomes are purified. But also for therapeutics, there are different type of purification. Yes, systems. But most of those purification systems that are good to use in therapeutics, most in therapeutics are based on size exclusion or size differentiation. Yes. So you just take one that part of all the extracellular vesicles, that group that have exactly the same size of the exosome. Yes.
Mm-hmm.
And in the lab, we have a technique that from here we can precipitate the exosome and purify more. Yes, that sample. But that is right now is not safe for human to use those exosomes
Why is it not safe?
because of the system, because of that system. You add things to the exosomes to precipitate. Yes, sometimes you detach that, but you have to use a lot of buffers, you have to wash them, you have to do, it's a hard process also. You can do that and now in science, but not, these are just for research. Yes.
Isn't also too like doesn't have like heavy metals? I believe we talked about or
Well,
something.
there are metal, yes. There are some precipitation and with
It is
magnets and
harmful.
that, yes. All that things. That is to have, and they are using more also maybe in one part of the diagnosis, because you just but you are when you in the diagnostic you to the sample, yes, blood sample for example from your patient, purify the exosomes, and you want to go a step further,
Okay.
you precipitate the exosomes, and after that you do the thing that you do is lyset the exosomes to do the studies. Yes, the studies on the cargo that you have to find the marker. And in that time, yes, you are in the lab. You are not going to use those exosomes for treatment. Yes, it's understood.
Yeah.
So, but right now for therapeutics, we are using size exclusion chromatography or tangential filtration. And what we obtain after that is just exosomes. Is a kind of purification or enrichment of exosomes, yes, from what the cell produce. When the cell produce them, you enrich that sample in exosomes, but you have other vesicles of the same size there. So all the exosomes that are around there, all the the products that are around there have exosomes and extracellular vesicles of the same size. Yes.
Interesting. Yeah. And then now these companies are just saying exosomes, but they get a mixture of EVs.
Well, that is the point that I wanted. If you want to go directly to that, it's a long talk. What they have there. we have to back again to the biology to answer, yes, that question. Is that, if you remember when we started the definition of exosome and the biogenesis, I say there is one specific one that are made from the endosome and specifically to transport the cargo, and the other vesicles that are from the membrane. And all those extracellular vesicles and other things that the cell secretes, other components, yes, other molecules that the cell secretes are in the media, are around the cell, yes, extracellular space. So there are companies that just and they put in the ingredients that just the product is just that extracellular medium that have everything. There are exosomes there, but they are more diluted in the middle in the middle of all this, yes, other components and other extracellular vesicles. Other companies that purify those exosomes, that do the tangential filtration or the size exclusion chromatography, and after that they quantify for the transmission electron microscopy and do the characterization if you have lucky, and after that you have there a enrichment of those exosomes, but there are another vesicles also of the same size. Yes. Yeah. Why that? Well, that is because, as I said before, we are running further than the technology. Yes, the nanotechnology. We are not yet there to really separate the exosomes exclusively from other vesicles of the same size with the technology that we have right now.
Wow.
Yes. So, but that will come. That will come.
Yeah.
I haven't. Yes. To be here, I was waiting 20 years. Everything comes.
Yeah. So are there any other misconceptions that you see on the
Yes.
market?
Oh, yes. Sure. Let me go, because there was with the cargo, I say the exosomes, the count. Yes. Well, the other, yes. Misconception. Well, maybe the other thing is more related with the product and the final product that we have. Let me think. Because I don't know if to talk about this in here. The other thing is, in relation of product, is what you add to the product. Of course, as we say, the source is important, and depending of the source, those exosomes will have a huge therapeutic potential, and also for regeneration and for anti-aging and all these things, depending of the source. Yes. Mm-hmm. But itself, the main thing is, and the big misconception is to treat the exosomes as another component, as another ingredient, as a molecule. Yes, and it's not. It's a, how can I say? It's an active agent. Yes, it's an active agent that have to maintain this structure, have to maintain the integrity of the membrane to really give the message. Yes, and for the cell to recognize them and really do the work. That the work in therapeutics is most specifically the regenerative, yes, the reprogramming function of the exosome, drug delivery, and that, and another. Also for cosmetic could be just the cargo, but for regenerative is the cargo and the reprogramming of the cell. So for the product that we have now in cosmetics, they mix and treat the exosome as another ingredient, and they mix for a bunch of things. And those bunch of things, as much as you add, also will be more difficult, yes, for the exosome to encounter the cell. And the other thing is that the cell have everything there. If you add more growth factors and more hyaluronic acid and all that thing, is like the cell is like full of things. So the best, that is a misconception that you can add, yes, lots of things, and that the exosome need, yes, for it, or more is like use the exosome as another ingredient. It's like using if you want to use exosomes, use the exosome as itself, and they will work very well. Of course, you have to really care about, in relation of what you want to treat, you have to be very careful in relation of the source that you choose. That's all.
Interesting. Okay. Is there any other myths that you wanted to share with us that you see out there in the market?
Will appear in our conversation.
Okay. Okay. I'm getting ahead of you.
Yeah.
Sorry. Okay.
Yeah.
Okay. So we touched a little bit about how when companies use the word exosomes on their products, like what are they often getting wrong? So from there, I know we touched that a little bit. They're getting a lot of things wrong, right? Is there anything else that you want to add to that?
Well, first is to put the name, yes, in the product, as we told, exosomes, without really characterize it. It's
Okay,
like,
so that's the first thing. So they should. So what
yeah.
do you suggest? Should they start just? So should they just use extracellular
Start.
vesicles?
I suggest. No, they can put exosome, but they need the characterization. They need the characterization, characterize them, and also we and the MISEV, we are going to add later on. We are thinking to add also the functionality, because to have a really intact membrane doesn't tell you nothing in relation of if that exosome will be functional or not. So you have to do the functionality analysis in relation of for what you will using it. Yes, that is also important. But what what is really misconception or no in relation of the name? Yes,
Yep.
the use. Well, I suppose that they put some companies that just collect the media. They say, well, yes, they are exosomes because I have the media and I know that there are exosomes in the media. Okay, yeah, there are exosomes, or other companies say, well, we purify them and we quantify in the nanosite. That is just particles per milliliter. So you to really talk about exosomes, you must characterize them. Now, also in MISEV, they are thinking just to talk about
Well,
until
Mazzeo,
the yes.
sorry to cut you off. I think it's important to mention who MISEV is.
Oh yeah.
Many people, I feel like, even including myself, before this, I didn't know who they were. So I think it would be very important for us to share who MISEV is.
Yes. MISEV is part, and I will say what that means. MISEV is part of the ISEV. ISEV is the International Society of Extracellular Vesicles, as I named it before. Inside that, there is a group of people. Well, most of that is open to all the scientific world. Yes, all
It's
the
public.
scientific
It's open
world that are,
publicly.
yes, that are no who participate. Who participate? I will say in this MISEV is most of the scientific world that are related with extracellular vesicles. Yes, so MISEV means minimal information for studies of extracellular vesicles, MISEV. Yes. So, and it's, yes, most of the scientific group participate in prepare those guidelines and come from our experience, yes, in the lab. Most of them are orientated for the studies in the scientific and the academia, not so much in relation of, and they struggle a lot, as I say, and I want to talk about that also in relation of the industry and how to produce them and the guidelines and the standards for that, for the scaling up, for in relation of the well, yes, the production, the stability. There are lot of topics that little by little start being part, because also they are creating the MISEV tech, because all the problems that now the industry has in scaling up, yes, the exosomes. But coming to the question that was, yes, in relation of the exosomes, they are what MISEV or is now telling us is until the technology improve to use more extracellular vesicles in general, because it's what at least at therapeutics is what we can have right now in the field. Yes, that are more. You have exosome, you enrich with exosomes, but most of them is mixed with other extracellular vesicles of the same size.
Okay. Yeah. I'm getting some great comments here.
Mm-hmm.
And I don't really want to read all of them, but one is that, the issue is how do we move from science to manufacturing and to
Yes.
clinical governance? You know,
Well, thank you for that question, because that was I was invited. I just come from London. I was invited to give a talk at the UK extracellular vesicles community, with other labs in the UK that are working on the MISEV, on the extracellular vesicles, most in the academia, yes, more
yeah,
science. And my topic was really in relation of that, how we go with what we have in science to the industry and establish those standards, yes, for the industry, most in relation of the scaling up, the reproducibility, and the conservation. That is also very controversial.
interesting.
Yes, and most because in all this, as I say, in all these steps, the main point is that if it's for therapeutics, how we preserve the potency. Yes, and in this for sure, and for therapeutics also we must of course characterize, but also do all the functionality and the potency analysis. Yes, that is the main point. But again, for the how the question was how we go. Yes,
How do we move from the science to manufacturing and to the clinical governance?
that is I'm coming back to the talk that I gave to them, because I was working in both sides, and I know the struggles in the industry. So I say to them, if you want really your amazing product, your amazing system, your amazing study, be a product later, yes, for a company, you really have to think, yes, on work together in how we can do this specifically, scaling up, thinking about the standard of pursuit in all the production system, how to maintain the reproducibility. The industry is doing something, but as I say, that may most of the time reduce the functionality and sometimes the function of the exosome. Like now they start to appear in relation of the reproducibility. Now start to appear those immortalized cells. The source are immortalized.
Yeah.
That is touching the DNA of the cell. So I really want to see that doing that change on the cells, you maintain exactly the same potency for that, and also the same cargo on the exosome, and there are nothing that affects also the safety, because also the safety concern. For that reason, the safety concern is something forward, and they are not using now on therapeutics because yes of the FDA concern in relation of that.
Yeah.
So that is something that is very challenging right now. How we are going to do this, and it's like the companies are trying to find a way. Yes, also for them to not make a very very expensive product. Yes. For example, now they are using in relation of the conservation, they are using the lyophilization, but the lyophilization, of course, that affects at some point the reprogramming part of the exosome. If you do that in a correct way, that is because you can do that and produce in a very restricted way, you can maintain, the best potentiality, that is not the same as the fresh one, of course.
Okay.
And the other is that change in a way the structure of the exosomes and how they are going, and from that how they are going to communicate with other cells. It's like when I think about that, I think if I want to conserve my eggs, yes, because I want to be mother later on, that are yes, freezed. Yes.
Yeah.
So they
Yeah.
are not lyophilized for some reason. So it's like lyophilization is used more for maintain the structure of components and that, but what is an exosome? We have to, if we use that, we have to be very, very, very careful and go to the expert. There is a way to do it, but you have to be very, very, very careful of the crystal that you are going to create to maintain the membrane. The membrane is the main part of the exosome to communicate, and most for therapeutics.
Interesting. I'm going a little off topic here, but I have a good question that I would like to ask.
Yes.
Is there a method to characterize exosomes like the characterization of growth factor related to the specific functions? Is there a method to characterize exosomes like the characterization of growth factor related to their specific functions?
Well, yes. That is the, as I say, that is what in MISEV we want to add the functionality. Yes, of course. Yes, that is something that must, yes, is and is in relation of all the whatever, the not only the functionality, but also you can do a proteomic and lipidomic. What I suggest to the companies is to know your product. Yes,
Yeah.
first the cell that they produce, and after that is the proteomic, the lipidomics, and the
Yeah, the biggest thing that I personally see, in my opinion, out there is that everyone's trying to make money with this, right? Because it's a hot topic, everyone wants it, and they cut corners and just to make money, right? Why
Well,
do you?
there are two things really for the working in a company that produces exosome is very challenging. Them the other thing I understand is, okay, we can make this product so expensive that nobody will buy. Yes, so they have the challenge in relation of the best exosome is nothing, just exosomes and just pure exosomes.
No extracellular
No,
vesicles, no other
no.
particles.
Well, no. Right now. I mean the product right now. The best one is the one that is not added nothing, and they are just not lyophilized. No,
Oh,
no. If you can have it, yes, not freeze it also, but you
yeah.
can freeze them and have.
Why do you think the terminology has become so inconsistent?
Well, it's because of that, because the misinformation, and
Yeah.
because it needed more education, and this that is a bit confusing topic. Yes, exosomes are confusing. You have to really understand this thing from the start: the biogenesis, the difference between other extracellular vesicles, the difference between different cells, and also in relation of the difference between their function and and how we can use them. Yes. Because that also changed not the way that we characterize them, but that's also changed the way that we are going to purify them. It's different, the technique that we can use when we do them for diagnostic purpose, that when used for therapeutic purpose. It's different, yes, because the process that we follow, yes, and the final product is different.
Yeah. Wow. So
that is confusing. Yes, that is
yeah,
confusing. For that reason, sometimes they say, "Oh yes, we and more for aesthetic. Well, if we are going to produce this, and if I ask how you characterize it, that, and they sometimes give me one technique that is really used for diagnostic, not for therapeutics, because yes, you are touching that functionality of exosome.
interesting. So
The other thing that came to me and I want to
yeah,
is, please, the company. Most of them give you the information from the exosomes and the characterization from the exosome before all the ingredients that are added. Well, if they give the characterization that added just to the product, but I want to see, and I try because I was testing lots of companies. Yes, in aesthetics, I was testing a lot of companies. I hope to do also functionality, not just the test. But and those that I found exosomes in the product, and the other is really it's needed to test the final product. Yes.
can you
Not
elaborate a little bit on that? Like, what does that, so people that are following, so the final products, because what are companies doing, or in your opinion, like, I mean, from your experience, what are companies doing? You said testing before, once they collect the product, they test it. So the numbers look phenomenal, and then they lyophilize it, or they do whatever, or add any products. And then it's finalized, and then they send it. Like, what is that difference? Like, how can it, obviously, I'm sure it'll lose its quality, its potency, everything, right?
yes,
So can
you
you,
have to test. You
yeah.
have to test
So
that. In
explain
the
it. Yeah.
final product, if you still have those exosomes and if you still um have that potency, well, we were talking more in therapeutic, but that is fundamental, because with most of the ingredients that they add, some of the in some products, because of the ingredient that they have, the membrane is just diluted. So you have something, and you have the results when you do your studies. Yes, after and before, because of course the cargo is there. But this reprogramming thing, the potency to do that.
Yeah. Okay. So so that's more on the therapeutic side. So I actually, we have a great question on by Emma. He said, "Hello from London. Nice to see you again, Carla."
Oh,
Uh,
Emma, yes.
she said, "What do you think is the biggest challenge with getting approval for an EV therapeutic?"
Safety,
Yeah.
safe, safe for human. I say that because, well, depend also from where your source. Yes, most the therapeutics right now, they are in some way modifying the source of the cell, the cell source. So that have to be, yes, to prove that is safety for human. That is the main point. And also, well, again insist the potency, to prove that during all the process, the production process, and the final product, when you test, when you characterize, when you test, when you do all the tests in the final product, please, is, and I told more, yes, for the products that right now for the cosmetic, and that the final product, that the exosome is there, and the potency. The potency is still there. And yes, the
Okay.
cargo that they have. Yes.
So
That is the main point.
okay. Yeah. Thank you for that. So what standards and or guidelines should companies be following when it comes to exosome products?
Well, I say what we have right now is the guidelines that we have from the MISEV. Yes. As I say, the minimum information for the studies of extracellular vesicles. Um, they give a lot of information step by step, but as I say, that is based more on the scientific studies rather than in industry. This is in process. That is the guidelines that we have. Well, that we have now is improving, as I say. The ISEV tech, yes, is coming, and we are meeting with. It's going to be a congress, I think in October. I have to check. So
In October
we are meeting.
2026.
We are all together. The people from almost from the industry that are in this field to figure out precisely, as I say, how you do the scaling, how you do the reproducibility and the conservation for the best of the exosomes, and if it's the best of the exosome, for the best of the patient. Yes,
Yeah.
to give the best that we have in relation of treatment to those patients. But as I say, the improvement of the technology, the nanotechnology is coming. We have to be patient. This is something that is coming.
Yeah. And, we can all agree that proper labeling, characterization is very important, not just in X, but in science and everything, right?
Everything, yeah.
What's at stake if we get that wrong? Like,
Well,
yeah,
that is in relation of um, again how you produce those exosomes. Yes, and also what I mean with that is that you really purify them, as we told before. What from where and how you obtain those exosomes, and in relation of that, be clear what you have in the product. That
yeah,
is the misconception. And also, well, as we said before, in relation of the concentration that you put, most of them is exosomes. Yes, to 100 billion of exosomes there. Well, be clear, yes, what we really have and the process, and be transparent with 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 information that we give to the client. Yes,
yeah,
in relation of how those exosomes were processed.
yeah,
That is fundamental, and from that come the misconception and all this, because they say, well, but this product has media, just cellular media.
yeah. And then are there anyone or any organization looking into that? Like regulatory, like bodies, like you said, MISEV is probably looking and setting and trying to set, well, trying no for a while, setting standards, right?
Yes.
Um, is there any, or do you see any progress in that?
Yes. There are a lot of uh, congress and meeting the industry also. But in this point, what I say is that yes, together we are, because I'm also talking with different companies that are not only for therapeutic, but also for medical device, from cargo delivery and other functions, all related, all industry related with exosomes. We try and we talk about how to put this in order, but also and it's good this meeting, the ISEV Tech. Okay, again, I'm insist is good because it's when the science also start to meet the industry, and this what I want to say and highlight is that this is a must. This is a must to work together in this and help each other and in this in this in this field, because
Yeah,
it's not easy, and well, and with again the technology that we have right now, we have to do the best that we have right now to provide that the best product
yeah,
that we can. There are other groups, and also myself. I really want to work on that, and I'm working with different labs here in Boston, in UK. There are a possibility in other parts of the world or Asia to work together, to really start to figure out about those standards mostly in industry, and to give that, and also to help companies in relation of their characterization or how to work, and the best, and also sterilize the production in general.
yeah. We have a question from Geely. She asked, "What are the recommended assays to accurately type and characterize the exosomes?" I believe we covered this a little bit at the very beginning.
Again, the question is,
What are the recommended assays to accurately type and characterize the exosome?
well, yes, the characterization, the main and the one that we are using right now is that we for to say that you have an exosomes in your sample, mixed with other vesicles or not, you must show that you have vesicles of the size of the exosomes, the biogenesis markers, those molecules that come from the biogenesis, and from the cell that gives them, from the biogenesis. And the other is the morphology, yes, for transmission electron microscopy. So proteomics, transmission electron microscopy, and nanotracking analysis.
That's why I wrote that at the very beginning. Okay, beautiful. Thank you. So if I'm a practitioner and I hear exosomes and I want to just start, using it on my patients,
Yeah.
what questions should I be asking vendors? I know I'm sure there's a lot,
Yeah.
but what are some basic questions that that we can ask?
Well, if you are a patient, as you are with your practitioner, or if you are a practitioner and you want to choose your exosomes to give, because that those are totally different.
Yes, I would say provider, because most of our audience, they're either physicians,
Okay.
and then
And
scientists.
they have the
Yeah.
patient that, so is the patient to the
So if I have my own practice, I'm a doctor, and I want to use exosomes, right?
okay.
And to my
Yes, you're the
patient,
doctor,
what are some good questions that I can ask the vendor or
the vendor. Perfect. Yes, now I'm okay. So yes, first the source, first question. Okay, from where those exosomes come from? First you need to really know. It's tricky, but you don't need to know for what you are going to use that. Yes,
Very
on
important.
the
Very important. Yes.
first you need to know for what.
Okay.
After that, to know which is your best source. Decide yourself. Decide, well, my best source, gold standard of this will be this.
Yeah.
So if you choose after that, yes, check the different vendors that you have, and ask them first the source of those exosomes. Second, the process that they do to purify them, if they purify them, which way and in which conditions, is it clinical grade or not, also important. Secondary, but important. And for the exosome, I say, no, for the patient. And the other is, yes, the characterization. Yes, to show those tests. And the other tests, yes, that are all the analytical from the cells and from the source, yes, that's fundamental.
Okay.
And apart from that is, well, whatever other component that is with the exosome, because yes, there are companies that add some components, other components there in the final product, but fundamental, the source. And the characterization, at least to know that they do that in the final product if it's possible.
And if you ask that to a vendor and they don't want to give it or they don't have it, that's a red flag, huh?
Yeah.
Run away.
Yeah. Yeah.
Run away as far as you possibly can. Okay.
Yeah. Yeah. At least, yes. Yeah. Yeah. You need to know from where those exosomes yes,
Do
where
you? Yeah.
it's like
You said
it's
you were testing exosomes. Did you, and I'm sure, did you call the company and ask them, hey, where's your COA?
yes,
Were
some
they?
yes. I have different things. One, the company gave me that directly, and the other was just as you say, the clinician that was, yes, wanting to know, okay, I really don't know which one to use, and I don't know what is here.
Yeah.
Like, what is here? So yeah, so I start to characterize them and
Did
try
you
to
have
identify.
did you have any did they give you any pushback when you started asking them these questions? Like, why do you want, did they, or were they easily giving you this information when you asked them?
Oh, that was lots of, yes, different.
Okay.
Some that is no, mine no, then well, okay, and
Those are
oh.
the ones that you need to probably get away from then, right? Those, right?
They have their reasons. So
Yeah. Okay. So,
yeah, but the good thing is that it's good for the companies that a third party characterize their exosomes, not they self, because it's like, yeah,
yeah,
if okay, I produce and characterize my own exosomes. It's good that you have a third party that really characterize your exosome and say, yes, you are right, you are producing the correct thing in the correct way. You have exosomes and
yeah.
they are.
Interesting. We have a very important, very interesting question here. Not sure if you can answer it, but the question is, how do you think EV dosage should be prescribed? For example, particles per kilogram, protein per kilogram, or any comments on this?
Partic as is the same as the um, in relation of the doses is tricky. Yes, it's tricky. We are still, yes, doing a study, because also it's in relation of the way of how you are going to administrate them. Yes,
That's true.
that change the concentration that you are going to use.
And going back to your answer to where you said, what is it for?
Exactly. You have to ask, well, what I'm going to use those exosomes. So the administration is one main point to see the concentration of them, but also is more relation with the frequency, also that relate with the way of administration, and also the for what, yes, the specific therapeutic issue, or also in aesthetic, the frequency also mark that. So from that is the main what we have done. The knowledge that we have, that is the particle per milliliter, because we measure them like that. There are some people that use lyophilized exosomes, and they have another standards for that in relation of the concentration. But for me, it's more in relation of the way of administration, in relation of how much exosome per milliliters you are going to add to give to the client of the patient, and this this way of administration and in this frequency.
Yeah. So what do you think the units of dosage should be?
The unit,
The unit.
well, the one that we have right now is particles per milliliter.
Okay, particles. That's what we use, huh? So per particle per milliliters. That makes sense. Okay, cool. So
Because most of, as I say, when you purify the exosome from the cell, is what you have. You have, yes, the exosomes in that uh, media or buffer that you have, yes, and also saline or whatever that you put, yes, your exosome when you start to concentrate and to purify them, and after that you measure in the nanotracking um, uh, in liquid, and they give the particle per milliliter.
okay. What are the biggest gaps in our current understanding of exosomes? The biggest gaps that you see in our current understanding of exosomes?
Well, first is what we told, what the information that we have, as I said, just right now, the what that means, the billions, yes, particles. That's or they say just exosome, but we know that we have a mix of vesicles there. But in relation of gaps, it's like one is, of course, this is the how we name there. The other is in relation of to use or to see, or again, not see the exosome as a active agent. Yes.
Active agent.
Now it's more see as a, well, whatever, another component that most for aesthetic, another component that we can, yes, add to the product that just right now exists, or with other formulation. Or the exosome, if you choose very very well your source, you can use the same the same product, yes, in different, for aesthetics, in different parts
Yep.
of your body, because it's going to be, if the source is good and the potency is high and cover a wide rejuvenation field, um, you are going to have a very very potent exosome that could, yes, be or act in whatever in whatever area you apply them.
Okay.
Don't need nothing more. They do the work.
Okay. So, Dr. Docukin, the leech doctor, says hi.
Oh yes, hi.
Yes. And one question that he asked, he says, are all current exosome products produced from human species?
Again, if the human,
If the
yes.
current exosome products produced today, are they all come from human species?
Oh no, not really. No, we have products, and most for regulation in Europe, those exosomes that come from plants. Yes.
Yep.
And also we have, well, also depend from what you are going to use them for. For example, for delivery, we have a bunch of vesicles. I say more vesicles. Exosomes also, but they are more general vesicles, like in relation of exosomes, they are using like the milk exosome for, yes, the liver drug. And also extracellular vesicles from bacteria. Bacteria doesn't have exosomes, so because it doesn't have the endosome pathway. And so they are using for also for delivery. They have also some benefits in aesthetics and some in therapeutic, but they are more used by drug delivery.
Yeah.
And yes, and most what is more now trending in Europe is the one that come from plants.
From plants.
But that is for aesthetics, for
Yeah,
therapeutics.
aesthetics, yeah.
As the human one, because the potency on that is again, that is more for using more that drug delivery than the reprogramming. Yes, part function of the exosome.
Okay. And then, yeah.
Well, content delivery, cargo delivery.
Yeah. What is from your experience, what is your take on using, let's say, autologous exosomes? And I'm sure derived from where, right? We all know that the source matters versus, a birth tissue neonatal exosomes.
From neonatal and Autologous. The thing is in the content, yes, the cargo that they have. When it's Autologous, well, those exosomes has your own age, your cells put there, yes. The amount of, yes.
And they can be stressed. So based on what we were talking about earlier, in their environment, if they were stressed or, in a bad
yes,
mood, then
all,
they can express bad
and
mood
also,
vibes.
yes, no, and also in relation
Yeah.
of, well, how can I say, all the bad things of the environment, yes.
Yeah.
How the environment were affecting you, yes. Though that is on all those exosomes. The only benefit in some way of Autologous is that you are, as I said, purifying and concentrated your own exosomes, yes, to treat one, I don't know, specific part
Indication.
of the, yes, and in your body, but it's not giving something extra,
Okay.
something more, yes, the potency of those that come and the wild that come from the stem cell from the neonatal, they are not affected. They were not affected from the environment, and they were perinatal really, yes, it's like so I'm not affected from the environment, and also there is an stem cell that really the cargo that is there is more wide, yes, it's more
Got it.
more concentrated. The potency is higher.
Okay.
Potency is higher.
Good. We have another question from Catherine. She asks, how do you assess the purity of exosomes slash EVs? How do you assess the purity of the exosomes slash EVs?
The purity, as I say, that depends on from what you are going to use those exosomes. If that is for diagnostic or if that is for therapeutics or drug delivery. As I say, until now, the way to purify and the way to enrich, yes, the product is through size exclusion chromatography, and we have right now different one. One is the tangential filtration, and the other is the size exclusion chromatography. And there is another one for a really good company that fasted the process of purification, combining the size exclusion chromatography with Cepharos with centrifugation. And right now is the most safest way to purify the exosomes.
Okay. So, what I see out in the market.
Again,
Oh,
I don't want the confusion. Purify, yes, the extracellular vesicles of the size of the exosome, enriched exosomes. I say purify exosome, I say, oh yes, purify. No. They are enrichment.
okay. What do you mean by enrichment?
Well, it's like if you have a bunch of particles, yes, in your sample,
Yeah.
and you have one specific one that is the one that you want. If you take out other thing that you don't want,
Mhm.
yes, the concentration of this in that yes, in the sample will be higher because you
Enriched.
take all the other things that was not part of
So
or not needed.
is enrichment the same as concentration?
Yes.
Okay.
Well,
What?
enrichment is go a bit further because concentration, you can concentrate all the molecules that you have there, yes,
Okay.
can I say, more concentrate with in the solution, but you enrich more if you take out things of that. Yes.
Okay. So
So
in a way, like
the
isolation.
number in the total number of the particles that you have, the number that you have there, yes, and in percentage increase.
Okay. Okay. So, in the market, what I see is a lot of, people using exosomes here for a lot of different applications. But there's also a lot of doctors, a lot of providers that are like, I don't want to touch it because it's not FDA approved or cleared, or, it's all over the place and there's no standard, right? That's why we're having this conversation, because we want to at least, learn myself. I want to learn and educate, having you, someone like yourself, to educate the industry so we can all move forward, right? What research still needs to be done before we can confidently use exosomes in a clinical application? Like, what does the research need to do in order before, so we can confidently use this in clinical settings?
Well, first is to, well, as I say again, the nanotechnology has to approve and to have the pure will be
So
great.
standardizing.
Yes, and also if we have the pure exosomes in our samples, but also we have to, as we say, we have to really do altogether the best job in the scaling up, and so how we are going to or we are doing now, but that has to be very, very standardized. How we are going to culture, yes,
Reproduce.
those cells that will produce the exosomes. Also in relation of how we are going to work on their reproducibility. Now, one of the, as I say, one of the things that they are using now is just one specimen source, like the cells that come from just one individual, and they do that, immortalize the cells that they are obtained, so they don't have to change the batch, yes, from the start. And again, do all the tests on that source. That really in which way that affect the final products and the potency is really need to check, and also if that change, yes, the content, the cargo that they deliver, that is important. So the main thing is the standards.
Would you say also that safety and like
Well,
studies?
that is number one for
Yeah.
therapeutics. That is number one.
Yeah.
The safety part, how they really relate with yes, in our body, and also that also comes from the source that
Yeah.
we use.
Of
Yeah,
course.
to be really sure in relation of
Okay.
this.
What excites you the most to where this field could go if we get the science right and everyone does their job by saying truth and labeling things the right way? What excites you the
Well,
most?
it's as for me, as I always say, it's the future. Exosomes could be the future of. I totally believe the future of the medicine. Um, and yes, for their function and for the potentiality that they have in therapeutics, in diagnostics, in all the things that we were discussing just today. So that is for me is fantastic. And this is a message that I want to give today, is that is fantastic, but depends of the the good things that we do today in relation with that, the way, depend of the way that we are now working in this field. Yes. Try to avoid of these misconceptions, to really educate in this topic that is it's challenging. It's not easy to understand how they work, etc. And also all the challenges that they have the industry now to have a final products on exosome. But that will come, as I say again. I repeat again. I love to be repetitive for things that are really important to
It can
recap.
get
Is
stuck in
that
your brain, you know?
yes? It's like the nanotechnology is improving. We are working that. My other message is we don't have to go, yes, faster than the technology that is coming. We just need to go good work, be open, and also, um, uh, well, um, say the truth, yes, of what we have,
Yeah.
what we are producing, what we and the way that we are producing, and share that.
Yeah.
Being open is the most way also for the science and for the technology. When I say be open, it's also to share the challenges that we have right now from one side, which is the science, and from one side, which is the industry. Yes, and yes, and between the
Yeah.
industry.
Yeah.
So that is the only way.
Yeah, and I agree because, for example, for me, like when I tried searching or, doing my own research on exosomes or any other, thing in science, I can't not find, maybe I'm looking in the wrong places, I don't know, but it's something to where we have, a good discourse, respectful discourse between scientists, doctors, and kind of go over what we see from clinical practice, from the lab to clinical practice. I think it's very important. And, I'm doing these things because I want to talk more. I want people, because I've learned so much just by talking to you and by talking to, everyone that I bring on the podcast. And my goal with this is that, you know, I see people talking about good topics in conferences, but
Mm,
it's challenging because not a lot of people, one, have the time or the money to travel to, to
mhm,
another city or another country to hear these types of information, right?
yeah.
In one of the comments today, I literally read, she's in school right now. She's in China. She's like, this helps me amazing. Thank you so much for having this, conversation. So when I read messages like that, it inspires me to continue doing more because there are people that are trying to learn and not trying to get into a conference, right? So
Yeah.
but it's just in the power of the internet. What you were mentioning about the science is moving faster than the technology, right? And it's the same thing we're experiencing with AI.
Yeah.
It's so fast, right? And I mean, there's going to be some bad things that may happen, but we're going to have to learn from that, right?
Yeah.
Um, so I would, yeah. And so that was just a little message from me.
Yeah.
All right.
I want to charge my phone. Just yes. I'm
No
still here.
problem.
Yeah. Yes, but
Okay.
tell me.
We cannot, I
Yeah.
cannot see you anymore though.
Yes, yes, but you can hear me, yes?
Yep, I can hear you.
Okay.
So
Yes, I'm
if
going
you
to
could send, if you could,
charge my mobile.
okay, if you could send one message, I'm not gonna be able. We're not gonna be able to see your face anymore, but it's okay.
No, no,
It's
no.
okay.
Soon, soon, soon. Wait, wait.
It's okay. No problem.
No, but yes, tell
So
me.
if yep, we got a couple of closing questions that I want to end with.
Yes.
If you could send one message to companies, to doctors, practitioners, what would it be?
Yes.
Let's start with
Yes.
companies first, I think.
It's like what I say before. My main point is that we need to work together. We need to really work together and and believe as I did when I started. Yes, um when we started sorry the noise when we started in this conversation that I was talking about how my beginnings with in the field of exosomes as well with my doctoral thesis on biogenesis of exosomes, and at that
Yeah.
point that nobody, well, most of the scientists don't believe on this thing called exosomes. So
Hmm.
so it's like believe in it.
Yeah.
Like I
I
did
mean,
before,
yeah.
things are coming. Um, uh, the technology is improving. Uh, we have to really be patient and and I say before, it's the same message is do the correct
Yeah.
thing. Here again.
Yeah.
Yeah. So so the main point is that we have to work together in this. The future of the exosome is fantastic.
Yeah.
All the if we work really well on it, uh, we can have a good diagnostics through them, good therapeutics, uh, and also I know laboratories that are working and preparing amazing drug deliveries based on the exosome. So I totally believe that if we do the correct thing right now, yes, and from the to go one by one is from the industry. Well, we have to work together and establish, yes, the standards, and maybe with myself and yeah,
Yeah. Well,
and also from the practitioners and from the clinicians is like to do the research first. Yes, know what your client needs, and after that do the research on what is the best source of exosomes you can give in relation of the treatment that you want to give to your client. So
um,
that is
yeah, I believe that everyone is listening to this, either live or will see it in the future. we're all believers of exosomes,
That's
right?
great.
Um, we're all believers. We've been going 2 hours, and I'll continue
I
more.
can continue.
Yeah, yeah, we can.
I have
Um,
more. I have more to say.
yeah.
So
So
I just resist myself. But
yeah.
yeah, we can continue another.
Well, yeah. Well,
Well,
I'll tell a little quick story, but I've met with Dr. Carla several times up in Boston, and when we meet, we've literally met and hung out for hours, like literally the whole day, you know, to where I ended up missing my train back because it was late. So my wife had to pick me up. So we lost track of time. We were in the lab, but
okay.
it's always a great time with Dr. Mazzeo for sure.
Okay. For me also. Yeah.
Yeah. So we have a question from someone that says
Yes, I'm interested in
if
those
we
those
have
questions.
if we have
I don't
a
know. I don't know if you can take those questions when
yeah,
after we finish. Okay,
okay,
because I
yeah,
if I
no,
tend to connect.
it goes with what we're discussing. That's why, like, when I read them, I
Yes,
pull them
yes,
up.
I
Yeah.
I'd like to know. I
And
see.
then one question, but it's that she asked Anna Maria, do you know of a list of companies that you found that offer real purified? I would like to maybe reach out to Dr. Mazzeo just because we want to avoid talking about a specific
Exactly.
company
Mhm.
or a
Yeah.
specific product. Maybe if they pay us, maybe we can talk about it probably, but not really, right? Right, Dr. Mazzeo?
Yeah.
But no,
Good.
just reach out to either myself or reach out to Dr. Mazzeo.
Yeah.
We can definitely direct you at least in the right direction. And then to answer that, Dr. Mazzeo put a good checklist, which is a vetting checklist as well. Now, I can go on and continue talking. Dr. Mazzeo can also continue talking. I have literally just two more questions for her, and that's going to be our closing for the conversation. But however, me and her, we asked each other if the audience wants to continue with us after we have this podcast, we can, and then we would like to go over the checklist. If that is okay with you guys, if you guys would like for us to continue specifically on the checklist, write on the comment saying yes, we would like to, or leave it for another day. It doesn't matter. We're gonna cover it either today or tomorrow. It's up to how you guys are feeling. So let us
Mhm.
know how you guys feel about that. Okay.
Yes, yes, that can be today or
Another
whatever
day. I
other
mean,
day that we
yeah. If
repeat
we
that
if you guys are
we focus exclusively on that checklist.
yeah,
Yeah.
yeah, yeah. We're gonna cover it, but okay. So my next question was, what resources or guidelines would you recommend for someone to who? What resources or guidelines would you recommend for someone who wants to learn the real science?
Yes, that is like a science exosome science secret. I don't
Oh,
know. But because I say, well, open to everybody, I love that. You had it, whatever you want to know, what the cargo of the exosomes on and in general, yes, exosome cargo. You can go to Exocart. That is one, or Vesiclepedia. Vesiclepedia has all the information in relation of different sources and different in general, from the sources and from the exosome cargo content. And the other is, well, of course, ISEF, MISEV. Yes, please read the MISEV. Um, uh, read the MISEV papers that are there, and those are the main sources that we have right now
okay,
to know more about the exosomes. There are, well, other more um those are more, yes.
okay. And then
The main information is there. Yeah.
and for those of you guys that have joined us after, MISEV stands for Minimal Information for Studies of Extracellular Vesicles. MISEV. Okay, and I'll share with you guys the website as well. if you guys reach out, and then it's actually on that checklist that we have created for you guys. Okay, so where can people, I've learned so much by Dr. Mazzeo, from
Uh,
Dr. Mazzeo, and I want to learn more. I want to follow you and see what you're up to.
yeah.
Where can they find you?
Well, for LinkedIn, yes,
LinkedIn? Okay. Yeah.
and the other is well, in Google Scholar. Also, Dr. Carla Mazzeo. You can find all my publications. Yes, all the publications that I have on the exosome field.
Okay.
And the other that I'm created is the Exosomes Guru that I'm in LinkedIn. Yes. Sorry, in Instagram.
And Instagram.
Yes, and in LinkedIn also I put yes, and I will share some information there. I'm starting to do
Okay.
that, to share things related more for the company view. And the other um well, yes, the other thing is like, well, now I'm creating, well, I do consultations, and also I'm creating this
Yeah.
safe company, safe TV company that is coming soon, and we will talk in a later conversation.
She has some exciting projects and things that she's working on.
Hmm.
Yes. And
So very big. So it takes
yeah. So,
time.
is before we end, is there any final message that you want to give to the exosome community, medical community?
And I also, well, open to, yes, to continue discussing, particularly someone have one specific
Yeah.
topic to discuss.
Okay.
Mhm.
Do you have any final message that you want to give before we log
Just
off?
continue doing good work. Yeah, that is the main thing. We're putting passion. That is the main thing. Um, and believe in this science, that together we can do it.
Together we can do anything.
Exactly.
Okay. Well, one last question I have for you, Dr. Mazzeo. Would you be, I know you're joining us from your cell phone. Would you be able to, do you have the checklist that we created in hand?
Yes,
Go over it, or do you
I
prefer that
think
we
I can.
cover that?
No, I can change. What?
Okay.
Sorry, I can change uh
What I would, because
to my laptop if you.
okay. Yeah, because I think it would probably be a good idea not to go, I think, too much into detail, I would say. But if we're going to be providing the audience with the checklist that you created, I think it might be a good opportunity to take this time to
Yeah.
just go over it and kind of talk a little bit about it, each section. So then, because what I want to do is I want to give you guys all the tools necessary, right? Like, I want you guys to walk away. Today is Sunday, and literally walk away tomorrow and be like, okay, I got a good
Okay,
checklist.
let's.
Let me see who's doing good exosome products, or because at the end of the day, we want to make sure that we are using safe products. At the end of
Exactly.
the
That
day,
is
for
that is that is the main. Yeah.
right, and so that's why I'm here to make sure, because again, I ask a lot of questions. Sometimes I can be annoying, and I get it. And I ask a lot too many questions, but I
But
think
I love that. So yeah,
yeah,
this, yes, to discuss about that, to see, well, how you yes, your point of view in relation of, yes,
yeah.
your
I
from
was
your experience, from my
yeah.
experience.
I think we all have, we can all learn from each other. I
Ex-
actually did while you are you looking for the checklist on your laptop?
Yes.
No, I'm just because I'm just killing time, but I wanted to share a quick story as we
Yes,
wait for
because
Dr.
everything
Mazzeo.
closed,
Yeah,
and yeah, and yes, that
yeah.
is yeah. But you can start. Yes. You can start with
Yeah,
the
I
check.
was just pretty much saying that I did an interview with
Yeah.
Dr. Alberto Gobi. He's from Italy. He's an orthopedic surgeon.
Oh, yes.
Yeah, and it was like a week ago or something like that that I did a podcast with him. And in our podcast, it's no, it's not on my YouTube channel yet. I will post it soon. We're just waiting on some things. But he told me that he told me, he's like, you are a teacher, Oscar. I've been following on your LinkedIn, and you ask a lot of questions, and you made us think. And I I reflected on that, and I was like, wow. I'm like, that sounds great to hear that, because I would have never known that a voice like me, because I'm not a doctor, I'm not a researcher, I'm just a person that has passionate for regenerative medicine, and I've been in the space for a decade. And so the fact that I heard that from a very well respected, you know, person
Mhm.
in medicine, made me realize that, yeah, like I don't have a lot of following, and I'm just beginning in my career. I would say I'm young, and the fact that that I heard that from him made me, like, inspired me to continue do more. I'm like, right, let me continue
Yeah.
to ask these questions.
Yes, exactly.
And so I just wanted to share that, because ask questions, because we don't know everything.
Hmm. Exactly. That is the way to learn.
So
It's the way to learn.
yeah,
No, but you are doing great in educating. Well, you give us also the space, yes, the
yes.
space to really.
Oh,
Uh,
yes. Yeah, I try. I mean, I'm not claiming to be an expert. I'm just trying. Okay, Dr. Mazzeo knows everything about. I'm not
well,
a lot
well,
about
I'm still,
exosomes, right?
I'm still
Um, More than anyone that
I'm never finished.
yeah, and
Because when you in science, most in science, when you yes, you have a question. You try to answer that question, and answering that question, another questions start to appear. So
more.
you never finish
Yeah,
because there is always a new
yeah.
question.
Okay. Now, regarding the checklist, do you want me? Well, I'm going to share the screen, because
Yes,
you will
perfect.
not be able.
I don't know. I have now is working. I don't know if you want me to go to the or just
Just keep it
continue with
like
my
that, I
one.
would say,
Okay.
yeah. And then I'll just share
Okay.
my screen, and then you can just
Yeah,
follow
I
along.
have in front of me. Yes.
Yeah. And then that way, my laptop. Okay, good.
Yes, I had that prepared everything, but after that, my
Yeah,
computer go.
yeah. And
Well, I have to
and
look
we
it.
logged in
Okay.
literally 2 hours before, to make sure that at my
Yeah,
check everything was ready. So,
yeah,
okay, Dr. Mazzeo.
it's
So,
there.
I'll let you take the wheel on this one.
No, me too. Yes, and I will. You ask the question and that,
Okay.
and I will yes help with the answers.
Okay, sure. So, this here is a document that I will be sharing with you guys. You guys can message me. I have a list of everyone that registered to the podcast interview. So I can send it to you guys after. If you guys are watching this on YouTube or on LinkedIn, either DM me, message me, email me, and I can send this over to you guys. Okay. So this was a checklist, a vendor checklist that Dr. Carla created. Um, Her new company is Standards and Assessments for Exosomes. She'll talk more about that. And then we separated this checklist by a few categories: product identity and characterization, the source and manufacturing, terminology and labeling, scientific evidence and claims, regulatory compliance, transparency, and then some red flags to avoid. And then for those of you guys that wanted the MISEV website, it's right at the bottom here. You guys can see it right here. Recommended resource, minimal information for studies of extracellular vesicles, MISEV. You click on that, you guys can have all the information needed there. Okay.
Hmm?
So I would say the first one is product identity and characterization here.
Yes.
So a credible vendor, right? We can all agree that a credible vendor should be able to tell you exactly what's in their product and how it was verified. If they don't,
Yeah.
run away, right? Can the vendor clearly describe if their product contained purified EVs or exosomes or just cellular medium? Can they verify that? What's your take on that, Dr. Carla?
Well, they really that is a must. You also from the vendor has to really know what is in the product, and what is in the product is, yes, from what we were talking, yes,
Okay.
to if you say if the product say we have exosomes in this quantity, well, okay. If you say that you have exosomes here, from which describe is a cellular media. If you or if you purify them, or if you enrich the exosomes, and you have that enrichment of exosome in the product, or it's just, yes, cellular media.
Okay.
That is a must. The company must.
So then you mentioned that there's three ways of characterizing an exosome, which is the nanoparticle tracking analysis, the electron microscopy, and then Western blot for exosome markers.
Yes, or that could be for the biogenesis marker. That could be whatever proteomics, but the most common is the Western blot.
Okay.
Mm-hmm. Yes, that's another must.
Another must. Okay. And then the vendor should provide a certificate of analysis with the particle size, concentration, and surface marker data. Are they able
Well,
to
yes,
provide
this is the characterization. Yes, sure.
that? Okay. And then here we go. Are the characterizations method aligned with MISEV, which is, again, minimal information for studies of extracellular vesicle guidelines? Shared with you guys at the bottom of that checklist, there's their website.
Mm-hmm.
Okay. How important is it that the certificate of analysis should come from a third-party lab?
Well, yes, we were talking a bit about that. It's for sure is better to have a third party that could do, yes, the characterization. Um, so that that avoid any conflict on that. Is a third party that certify that, yes, that you have exosome in your product. So that is another, if you, yes, if the company can afford that, it's a must also.
Okay. So going over to the source and manufacturing, we can all agree, right, that the origin and the production process it directly impacts patient safety, potency, consistency, functionality. I would say, right, literally everything. So does the vendor disclose? So a question you should be asking is, does the vendor disclose the biological source of the exosome? How important is that, Dr. Mazzeo?
Yes, it's totally another must.
Yeah.
The source of the exosomes, as we were talking, that is depend of what they are going to communicate to other cells, and that depend of the source that we obtain those exosomes,
Yeah.
the source that produces those exosomes. So yeah.
Good. So the vendor should disclose the cell source, traceability, and then documented donor screening.
Mm-hmm.
That should also
Yes.
be on
Also,
there.
yeah. Yeah. Mm-hmm.
Does the vendor also describe their manufacturing and isolation processes? So what did they do? How did they process it? How important is that?
Um, Well, they have to describe, as we say, most in relation of the enrichment of the, yes, the purification, how they purify those exosomes is important. Yeah, that is in the quality and affect later the potency. So that is the main.
Gotcha. Does that
Mhm.
also that should be included in the certificate of analysis?
Not
No.
necessarily.
Okay.
But if you, as um, as a client, yes, of this company, ask, okay, I want to know how you proceed, how you do this, they have to give that information. Yeah, sure. Hmm.
Okay. So the
Um,
product obviously should be, I know the FDA is very strict when it comes to GMP, sorry, good manufacturing process. Uh, that should also be obviously taken very seriously by the lab and whoever's manufacturing these exosomes.
yes.
Um, our batch to batch consistency and quality control measures documents. We put that information on there. Good luck getting this from the company. Um,
Yes. Yes, because that is also it's a challenging. Yeah.
yeah, and I actually asked you this when we were getting ready and kind of going over everything what we were going to be discussing, because and I'm sure it costs probably the lab and the manufacturer a lot of money, right, for them to batch test every batch.
Yeah.
Um,
Yes. Well, for that reason now, yes, companies are talking about the immortalized.
what does that mean? Immortalize it. What does that mean?
Well, there because we didn't talk about that. There are lots still to talk. There are a lot. So, but really, this is more from stem cells that there is a specific number of passages that you do for the stem cell maintain that condition of a stem. Yes. So,
Mhm.
if not, they start to differentiate. So the thing is that after these passages, the time that you that you reach those passages, you have to change, yes, the entire batch, and you have to start again all the process of test and and etc. for the new batch. So that is something that sorry, so that is something that the companies try to avoid because that is also time consuming and also money. So what they start to do is to, from one great donor that have amazing, yes, produce amazing, or they can extract amazing cells to produce a good quantity of exosomes.
Yeah.
What they do is to um, um, change that cells, yes, change that cells and make them um, to reproduce um, without senescence or without getting old, and you can have more passages, more than four or that, because they maintain exactly the same condition and the same state. So that is, yeah,
Important.
the but yes, but the thing is that how that affects the cargo. Yes, they have to prove that the exosome are exactly the same as the same cell, um, the naive cell. Yes, without touching that cell, what the naive cell secrete without that modification.
In a perfect world, do you think it would be ideal for a company to show that, like from the batch to batch consistency?
Yeah.
Yeah, I mean, of
Yes.
course, right?
Yes. It's just, yes. It's just well, to put work and time on, and to, yes, and to do the things correctly and all together design those, yes, all the standards that we need, and also a relation of that. Yeah.
I agree.
Mm, yeah.
Another question is on this checklist. If you guys are just logging on, I'll be sending, for those of you guys that registered, I'll send you guys this checklist. If you're watching it on LinkedIn, because I saw a couple of people join, message me and I can send you guys this PDF doc over here. Does the vendor test for sterility, endotoxins, and other contaminants? It's a huge one as well.
Yeah. That is, yes, that is part of in the certificate on conformity to have all those. all the tests. That's sure, and that is what I think. If you have just one donor and you modify those cells, you don't have to proceed again and again to check all the things all the time. Well, you have to do with other kind, like the media that you use and all that thing, but on the cell, yeah.
Okay. This question here, does the vendor, so now we're going into the terminology and labeling, which is, very important for us to obviously move forward, use the correct terms. Does the vendor use scientifically accurate terminology consistent with the MISEV standards? What does that mean?
Well, that means that if you, as we say before, it's in relation of if you say that you purify the exosomes, yes, and you have enrichment exosome in your sample, is the well, you can use the word exosomes, yes, and the product. But if they are not purified, well, maybe it's better to use extracellular vesicles.
Okay. So now, if the next one is if the product is labeled as exosome,
Sorry, or and media more. And media, extracellular vesicles, or media, yeah, cell media, if it's what to put there, what it is, and that give out information of what was made in relation of that the exosome, yes, if they were purified or not, or all that information.
or using the general cell medium, which is what you were saying, like it has been
Mhm.
purified. And based on, I know you covered it at the beginning of this conversation, you were saying that there are companies out there that are selling that medium, and it's just a whole bunch of diluted
Mhm.
product. That's kind of what I understood from how you explained it to us.
Well, yes, it's just the media where the cells were growing. Um, with all the other, not just the extracellular vesicles, but other components that the cell secretes. So that is the extracellular media. Yeah.
So if you see a company saying exosomes highly concentrated, pure exosomes, what's
Well, now we know that is not the case. They enrich the sample, but they are not pure.
interesting? Interesting. So what would you advise them to say, right? I mean, because obviously, I believe that's a marketing terminology or a marketing tactic.
Well, not to say not to say pure. Like if they add exosomes in the label, just percent apart, yes, all the with the product or some possibility with the web page or whatever, that the clients can go there and see all the like this, all the check that they do the work, they characterize how they purify the exosome, if they enrich or not, and what is exactly in that sample, and of course all the tests on the source of the product. That is fundamental.
Okay. So another checklist here item is, does the label accurately reflects the contents, including the source material and isolation method? So the documentation should include the source material and isolation. What would that look like? Like from the source material, what would be an example to that?
From the source.
Yeah, like how would
As-
it say? Like this is from human derived? Like how would
yes, exactly. And also you test also from the source. If that, for example, if that come from, I don't know, from umbilical cord, for example, well, okay, you have to test. You have all the tests that were made also from the mother. So all those tests. That is for safety. That is all related also for safety. That is the main point also.
yeah,
Yeah,
that is the main
for
point.
the therapeutic more. Yeah.
Okay, so we touched a little bit up here where it says stem cell exosomes or pure exosomes. These are some marketing language as we discussed.
Mm-hmm.
What type of data would support that claim, let's say? Well, yeah.
Uh,
So if
what is
you,
that?
yeah, so for example, if you see on a marketing brochure, let's say pure exosome, right? Like,
you are in that. Yes.
yep.
No. I saw that you went to the other question, and I
It's
say,
football.
sorry. Yes. No. In relation of that is yes, of course. Well, you said stem cell exosome. Okay, stem cell. Okay, describe your stem cell, your source. Yeah. Okay. Yes. Everything has to be yes.
Documented, supported.
Exactly. That is the only way that we can do good on this. Is just to tell, yes, to educate yourself and put there
Yeah.
all the information.
Yeah. So let's pivot to scientific evidence and claims. So this one right here, it says, does the vendor provide peer-reviewed published research supporting their specific product, right? We the way that exosomes are, I mean, yes, they've been studied, but how for a provider that is getting into that is learning into exosomes and all that, a lot of them show me the data. I mean, is there enough data for them, to support? There's none. So how can someone make a good judgment because there's not much published? Sorry, let's go back.
Yeah.
There's not that much published or peer reviewed data out there. So how can they make a good judgment on that?
Well, again, talking with the company that produced those exosomes, trying to find as much information that you have on how those exosomes were produced. If they do also, well, it's that is most an aesthetic. It's not say too much if they just do studies after and before. This is not the main point, but yes, if they do some apart from that functional studies. And yes, that will be great if the company works with academy and they also test or do research on that specific products and they can share that that those peer reviewer yes um, journals or what call papers, yes.
Okay.
So to support that, but most is for the to have access of how the company produce that product. Yeah.
Okay. I know that.
Right now, this will well, as I say,
Yeah.
the technology will improve again. I say so. We have to be patient. Also, that will come.
Yeah. So I know this next one is a huge one, is a big one. And you did touch on it. Are the studies or the white paper, because I know some companies provide white papers on their own product, right? Are they conducted and actually on the actual products being sold? So meaning from like from before they got lyophilized or as soon as they retrieved or got that product. Can you elaborate a little bit on this one a little bit?
Well, yes, this question is when all the studies that they conducted, if that is on the exosomes that they have in the product or the final product. That is because there are some products there that they as we talk a bit during our conversation is that some companies start to add things yes in the final product. So the final product is not just extracellular vesicles or or purified exosomes. They have more things there. So the thing is that if the company or the studies that they provide is in the final products or the exosome before to have the final product, then it's a huge difference because, as I say, with all the things that you add, the functionality or the potency could change. And also, yes, and also in some products, you can find exosomes in the final product. Yes, after adding all the other components that are part of of the formulation of the ingredients. So that is the main point.
Well,
and the same with the lyophilized exosome. Most of the studies are made are related with the products. Most are made before the exosomes were lyophilized. But what happened when these exosomes were lyophilized and also mixed with other things that are not lyophilized anymore, because all the things that they introduce in the as part of the product. So the main point is that they can or not really repurify the exosome from the final product, yes or not. And those exosomes that you repurify from the final products, those are maintain the functionality and the potency.
Okay. How important is it that the vendor distinguishes in vitro versus animal and human? Obviously, it's super important. How?
Well, all those, yes, are important.
Yeah.
But there are, well, most on in vitro findings and in animal studies, there are more in relation, as we say, in relation of the potency and that. Well, in human clinical, well, I was
Well, it should be. Well, it depends. So whatever, for whatever reason you're going to be using the exosome for, either in a
well,
lab
that
setting,
is
that
the main
makes, yeah, yeah,
fundamental. If
yeah.
you are talking, yes, about the therapeutics, of if you are going about aesthetics, this is a bit different. But of course, all the tests in the lab, like as I say, in vitro findings, and well, in aesthetic, the animal studies. But in vitro more, yes, we can do for that the functionality analysis, and this again is start to be a must also to show that the exosomes that are in the final products are really functional. Not just on the studies on the clients or on the patient, the after and before, but the specific studies, functional studies. That is important.
Okay. This one question here: is the vendor transparent about the limitations of current research and what remains unknown? From your experience, do you think the vendor is aware and up to date with the research? And they should be, right? But are they?
Yes, well, this is again more education. Yes, this is just to and in a way to really understand this science. That is also fundamental, because that is the vendor has to explain, yes, how the product works, and the vendor doesn't have this real information.
Yeah,
That is where all this confusion starts.
that's true.
So
I mean,
it's good to go um to the what can I say, to the person that have the knowledge, yes, and really understand, yes, the science. Mm-hmm.
okay. Now, pivoting to the regulatory compliance, we know that exosomes and are still evolving. The FDA has issued several statements claiming that, obviously they're not FDA approved for anything that I know of, I believe, as of today. Um,
Yeah.
and companies doing the right way, the right things, the right way. Uh, I mean, if I were to be an exosome manufacturer, I would be doing things the right way, right? I mean, some don't, I guess. But
Yes,
does
that
the
is.
vendor have FDA registration? And the FDA registration is you can go to the FDA, the government website, and you can find out if that vendor is registered
Well,
with
but
the
this is again, this is in relation of is for cosmetics or aesthetics, or is something that is used as a therapeutics, and also the way that you
FDA.
apply that is totally different. You apply
That's
in
true.
your body. Um, for that reason, in relation of cosmetics and aesthetics, is if you are applied topical, that the FDA is not have to be regulated, but that is not regulated for the FDA. The thing is, but with the therapeutics, well, there are some trials there, and is coming. It's all related of the research that we are doing now in relation of the safety of this.
Yes. Well, do you think that because you mentioned a good point, this is more for therapeutic, but then the FDA and this is not supposed to be, I'm not an expert in regulatory by any means. I just want to say that out there. I'm just learning as I go, right? So, um, do your own research is what I'm pretty much saying. Well, from what I know on the aesthetic side, because it's not, let's say, enforced or because it's used topical or whatever, I'm sure there's a lot of companies that are cutting corners and
To
that are cutting corners, meaning that they're not doing things the right way,
oh yeah
right? To either
yeah
save them money, or to make more money, right? So I believe, in my opinion, is that even if you are if you want to be here, 5, 10, 20 plus years, I would say that do the things that the FDA requires, even if it's not enforced. I mean, at least that's my opinion. Um, but yes, for the companies that are more for the therapeutic side, yes. Warning letters. What is your opinion on warning letters, and have they resolved it?
No, yes, I think you have to check if your company, because there are some exosome production companies that has warning letters. So
Yeah,
because they were producing in in a correct way, or they are applying them, yes, or selling them not in the correct way. So they have these warnings, and yes, you have to check for that.
yeah.
Mm.
Okay. Um, do another thing to make sure, does the vendor proactively follow evolving regulations and communicate changes to their consumers? Obviously, if anything happens, I think it would be, their responsibility to.
Exactly, to inform.
Mhm,
Yes,
mhm.
totally.
Yes. From your experience, is the product marketed with appropriate disclaimers and without unapproved therapeutic claims? I see so many companies making these claims and not marketing properly. I mean, I would say that most FDA warning letters, I would say, are marketing claims that
Yeah,
they are saying.
exactly. Yeah, those are. Yeah.
Yeah, right. I would say there are some that are maybe good manufacturing processing, right? That where they're not practicing or abiding by those guidelines, I would say. But I would say most of them are marketing things that they cannot say.
Well, yes, it's totally different. And if you are the language, and the companies know that the language that you use for cosmetics or aesthetics, and for therapeutics
Different.
and regenerative is totally different. The language that you have to use, because you can't say that you treat something if you are in aesthetics.
Oh, they'll come after you fast.
Yeah, no, you can. It's not, you are not treating, because you are not using this for therapeutics exclusively. Yes, you help your skin to look better, and yes, but not treating.
Right.
And like that, yes, there is a all totally different vocabulary that you have to use for,
Um,
yes, to
okay.
avoid.
Yeah, I want to add something to that. So I forgot who I was speaking to. We're
Hmm.
talking about hiring an attorney or hiring someone that knows regulatory and compliance. And we were talking about it. I forgot who it was. I wish I could remember who it was. And she made a good point. And the point that she made was, but attorneys don't even know the language because it's new.
Yeah.
Like the attorneys and like we're all trying to figure it out what is right, right? And then how do
No,
we?
but not in relation of the exosome, but they know, and they can help which which language, how to
Yeah.
yes, how to communicate in the
Yeah.
aesthetics world and relation
Yeah.
of the therapeutics. They are specific. That doesn't have to be specifically with the exosome, but it's in general.
Yeah.
Yes, there are specific. You can relate nothing. You just you can say nothing that could relate with therapeutics or with the treatment.
Yeah. Yeah. Because it has to go through clinical trials and be able to
Mm,
market it as such, right?
mhm.
Um, Next one is transparency and customer support. So a trusty vendor, we can all agree that they will welcome questions, no hiding anything, and here's my documentation, right?
Mhm.
Um, what is a technical documentation and SOPs? What specifically is that?
Well, that is not something. Well, have to be there. It's not something that they really have to. Well, the standard of pursuit and the technical communication, full technical communication. We talk what the vendor had to have, which is all the tests, yes, on the source and all that things. The full technical have to be there. Yes, you have to have access of that. It's not something that they must have, but at least the possibility to reach that is yes, it's a question of reach yes from the clients, or it's like okay, we can reach this information and have the answer easily and not yes
Gotcha.
go through yeah.
So essentially,
Uh,
you'll have to. So essentially, you'll have to call the vendor and the company and say, "Hey, I'm
ex-
requesting this," and they should be able to. If from your experience, did you find it hard or difficult or challenging
from
for them to
yeah
provide that information? Like, why do you want this information? Right? Like, yeah,
yeah
yeah, they gave you pushback. Well, if for example, me, if I were to be if someone were to be asking me that, if I was a vendor or I owned an exosome company or whatever, and someone would ask me that, I guess I can understand why I'm a little like pushback or like why I would push back. Like, why do you want that? Because don't you think that
yeah,
other
you start to say well something weird yeah
yeah,
yeah.
or like another company is trying to steal my processes, right? So that can be another challenge of why they don't.
Well, that will change when everything become more standardized and yeah.
That's true. That's true.
Mhm.
Okay. The next one is, does a vendor have qualified scientific personnel available to answer any technical questions? I mean, I would say for the most part, most vendors do have a a scientific director or a scientist like yourself in
Yeah,
the,
but especially that not really the product and not really the exosome field. Yes, that is the main point. Most because most because of what of well as I say this is not an easy topic to understand also for scientists. That new on this field. So so to have someone that have yes experience and more as scientist that has experience
yeah.
on exosomes and yeah
Okay.
that to answer every
Question.
technical and scientific question
Yeah.
that is
Yeah.
good.
So another important thing is that, does the vendor offer training or education on proper handling, storage, administration on the product? This is more on the therapeutic side. Um,
Oh no, also cosmetics. They have some education. I know companies that have education and training courses for their staff and the vendors and that. So and that is fantastic
yeah.
because they give yes, they are good education for them.
That is, I mean, most of the physicians or the providers that reach out to me is that same question. Is like, oh, do you have education or proper or support, right? Because
Yeah, that's fundamental.
it's fundamental. I mean,
But that is that what I mean is fundamental is because that is where all the misinformation start to appear, and after that nobody understands nothing about what is an exosome. It's like So this is fundamental because of that. So to avoid all the misinterpretation, all the misinformation that are now around.
Okay. Next one is the storage requirements, shelf life and handling instructions clearly provided. From your experience, or were they providing this information with the companies that you tested? Most of them?
Not all you can find. Yes. Different things. Not all really. Not all give you all the how. Yes, how you can handle what's happened. Well, first in the how they are delivered and how this product is, or the exosomes in these products are conserved, and after that, okay, what you have to do when you receive this specific sample. Yes, have to go directly to the freezer, to the
How to thaw it,
fridge. Yes,
how to reconstitute it if you need to, right? Some of them you have to.
in which time, and also lot of question for the clinician is that, well, if I use just a bit, can I keep the rest of the products apart and to freeze again or put in the fridge again? So all those questions, yes, how to handle that product when the client receive, that is something that had to be there, or at least for the clients easily access that information.
Okay.
Mhm. Yeah.
And then some exosome products, correct me if I'm wrong, they have to be reconstituted too, right? Some of them, or no?
No, just if they are lyophilized.
Okay. Oh, okay.
That is when you reconstitute. For that reason,
What
that's
are they
also the
reconstituted?
way that you reconstitute. Also, you have to be careful, and yeah, sometimes you mess up all
Interesting.
the product.
Yeah, I mean, and yeah, I can see that happening a lot. I mean, with PRP, I see that a lot.
Hmm.
Okay. Um, does the vendor track and report adverse events or reactions or product complaints? They should, but do they?
Well, as you say, I they sure, yes, they must be. This is something that, well, for therapeutic for sure, this is something that must to be there, accessible for the patient, but also for aesthetics will be good to have this kind of information on this. Not maybe not from the not directly from the companies, but some other organization that really warning, yes, the clients that, okay, they have this and this. Yes, that information has to be there in some way.
Okay.
Hmm.
So here are some red flags, and that I believe that Dr. Mazzeo and believes that should proceed with caution. If they
Yeah.
cannot provide a COA, run away.
Hmm.
That's a pretty good tagline. If they don't provide a COA, run away.
Yeah.
I'm gonna trademark that. Product labeling uses vague or scientifically inaccurate terms without supporting data.
Exactly. That is. Hmm.
So making claims, pure exosomes. Would
With
you say,
characteristics.
yeah, would you say that a red flag would also be if a company says 1 trillion exosomes in this vial, or 1 or 10 billion, whatever, if they put a number, would you consider that a red flag?
Well, the correct way to say that is as the machine gives you, yes, it's particle per milliliter, and after that to say, well, we purify the exosomes the exosomes or the extracellular vesicles in this way to enrich on exosomes. That will be the correct way.
So companies that are providing numbers, is that a would you say that's a marketing
Yes,
tactic?
they provide the quantification, yes, the number.
Oh, so would
Because
you So my question is, if a company is giving a number, right? Because is that okay?
Yeah.
Okay.
Yes, but they have to say what exactly those numbers. What I mean is that they have to really highlight what those numbers means.
Yeah.
That's because if you don't say nothing, it's like you have exactly that number as pure exosomes. It's
Yeah.
not true.
And then actually touching on that topic, I've also seen companies that they sell different types of volumes or concentration. So for example, the tier 1 is, I don't know, 1 trillion exosomes. The next one is 2 trillion exosomes. And then the more expensive one is 3 trillion exosomes. Does
Yes,
that
but
make
this
sense?
is no, no, no. Well, yes, you can have as much exosome. It's also depend again. It's also depend how you are going to use those exosomes. Yes. It's not that, for example, I don't know, if you want to apply that in your face for the wrinkles or, um, um, yes, for cosmetics, it's not that as much as you apply, you will have the best results. Yes.
Yeah.
More exosome better. No, it's not like that, because this is, as I say, this is not another component or ingredient that you add there. This is an active agent, and they have to reach the receptor cell, and they do something, yes, with that receptor cell, and they have to really,
Yep.
and from the receptor cell, apart from recognize them, this cell also could become saturated. Yes, they just have a range of how is called uh, a limit, yes, of how much they can to receive. So it's not like as much, yes.
Okay.
Better result that could cover and reach more cells. Yes, but maybe with less concentration, you have the potency is higher.
Okay. So another red flag is if they don't disclose the biological source or the cell type or isolation method.
Sorry, from you lose them.
Yep. I said another red flag is if they don't if they do not show or disclose of the biological source, cell type, and isolation.
Oh yeah, that is, yes. I put that is, of course, the main thing. If you don't know from where they come, you don't know nothing of what you have there. Yeah.
Run away.
Mhm.
The therapeutic claims. Obviously, they're saying this cures anti-aging, but let's say, I mean, maybe a company. I mean, I would say yes, they are not compliant, right? By if they are making claims. Would you say that? I mean, it is a red flag because that tells me that they're not doing their homework properly, right? They're
Hmm.
not. They're new because a real company would know, hopefully, that you can't make any claims, right?
Yeah, well, but also from the therapeutic, yes. But there are not, well, you can use in most of here as a therapeutic. Most for, well, yes, this is for another conversation later.
Yeah,
But yes, all has to be based on the clinical evidence, of
yeah,
course. Most
yeah.
for therapeutics, most, yeah.
Another red flag is if they don't have evidence of good manufacturing processes
Mhm.
or conditions.
Exactly.
Huge.
Yes.
Cited studies are not specific to the product being sold or rely solely on generic exosome literature. But the way that I see this, for example, is remember that we mentioned earlier in this conversation to where there's not many clinical studies or studies about exosomes, right? So how can a vendor or a company prove, right, that the studies is
Well, from the
safe and
well,
effective?
well, they can provide, as I say, the potency analysis, and yes, is to do not yet on human, or they can start to, yes, try to start a trial, but before that you have to really prove that your exosomes have the potency to act in the therapeutic field that you want to use them. So
Yeah.
that is from start. It's like before to go, yes, to other things and to trials and to other kind of studies. You have to really prove that your exosome have the potency that you reclaim. Yeah, that's for sure. And you can do that on the lab.
Yep. A huge red flag would be if the vendor has an unresolved FDA warning letter or a history of non-compliance. That's huge.
Mm-hmm.
If they haven't answered to the FDA, that's a big red flag.
Exactly.
What else are they not answering to or cutting corners, right?
Well, that for sure. The thing is that not sometimes it's not easy to find that, but yes, that
The message gets across relatively quick. I feel like, especially if you're talking about exosomes. I feel in this industry, people will make it known when it's companies get a warning letter. There's a lot of people out there. So another red flag is that they don't have a scientist or a scientific staff available to answer questions. But I've seen it to where they don't.
hmm. Yes, most that come most for there are some companies that they sorry. Oh, okay.
Salud.
That yeah. Um that they buy the exosome for a production company, and after that they create or formulate their product with the exosomes that a production exosome company provide them. Yeah.
Yeah.
So yes, they know about, well, the products that they want to commercialize, but not in relation of maybe the specific or how those exosomes were produced. Maybe yes, they know and they have all the tests and everything from the source of those exosomes, but maybe not how the company that provide those exosomes were produced. That happened, and that is, well, the company that really sell the product is where they don't have the scientific knowledge, and specifically on the exosomes, because they trust on them, on the company where they bought or they provide them the exosomes.
Yeah. Well, I mean, you are 100% right. That's everyone is relying on the company.
Mm-hmm.
I would say the providers and the people that are not aware and really do not fully understand the regulatory aspect and the science, I would feel that they are in that bucket.
Mm-hmm.
But for the ones that that are aware and that realize that the exosome industry is just not easy or simple as some people say they are,
Yeah.
not right. Um,
Yeah.
okay, then another red flag is reliance on testimonials, influencers. Well, obviously, we see that a lot, especially on social media, right? With influencers saying that
Lots.
I regrew everything my hair. I mean, even
Well,
though it may be true, my
yes.
hair and all of that, and then before and after photos as a primary evidence. I mean, yeah, you were going to say something, Dr. Mazzeo. I'm sorry.
No, yes, in this specific, you start, yes, I start yeah, to remember things in relation of this, that most from that come from different congress in aesthetics that's um I was participating, and the main thing was that the the client insists to the practitioner, the clinician, that I want this because I see that, and the clinician is like, or maybe they know about the exosome and they know the goes and pros on using that, and it's like the insistence of the of the clients that they want that because they just, and that is also
And I want to add.
that is where the thing start to
Yeah, I want to add something to that because you made a very good point, and I actually had a personal experience on that. And so, for those of you guys that don't know, I built I created a free online course that covers the FDA guidelines on unapproved drugs, right? So it covers, it's in our website, regenmedacademy.com. And all it is, it goes over the expanded access program, the right to try. It goes over data integrity. It goes over also compliant marketing. And then the Chevron deference. With the Chevron deference is something that happened that was overturned by Congress. You guys should definitely look that up because it impacts healthcare and the laws and stuff like that. And so as I was building this course, I was reaching out to manufacturers in the space. And one question I introduced, she's like, "Oh my God, this is amazing." She said, we want to use this. We want to give this to our vendors, to our distributors, to our team and everything. I'm like, okay, awesome. And then I asked her one specific question. From the people that just want the products, because what you said, there are doctors that say, "I just want the product, give them to me." And then I said, those people that just want the products and they don't want to understand the science, the regulatory, or whatever that is, what type of clients or customers are they? And she said, we don't like working with them. They are the ones that produce the biggest problems or the issues for us. And I asked, why? Because those are the ones that are not compliant. Those are the ones that are saying specific things, right? That are making claims on their website, on their marketing brochures. And what happens? It all goes back to the manufacturer.
Yes.
I just wanted to bring that up because you mentioned, and I reminded me of that story.
Yes, that's happened. I was there, and that is for some practitioners it's a big problem.
Yeah. And then one thing that I do want to add specifically on the heavy reliance of testimonial influencer before and after photos of primary evidence. So it says primary evidence. There is, and I see this a lot because we do marketing for a lot of regenerative medicine clinics and tech, biotechnology companies and distributors. And it's okay to use before and after pictures. That's okay. However, you need to add a disclaimer at the bottom or somewhere that says outcomes and results may vary. Because in a way, by adding a disclaimer thing, because it's true, if I get an exosome or whatever, and it might have the outcomes or the results might be different than from you, from me, for someone else, right? So if you're going to be using before and after pictures, this is more on the marketing side, make sure that you are using disclaimers and saying that results may vary, right? It's like in the casinos, right? When you see the things for the casino or even pharmaceutical companies, when they start talking about this product does this, what do you see at the bottom? The disclaimer, right? So it's okay to use it and we should use it. I think it's great, but disclaimers is important.
Yes. Well, that also refers to but also from the characteristic of the clients and the batch to batch consistency. Yes, when you produce the exosome, both things have to come together to have at least similar results.
Yeah.
So that is also important. Both things. And also some companies also confuse what is how it's called, the the word is doesn't come. Well, the after and before and the potency, which is totally different studies and totally different things. One you do on the clients and is what's happening on the clients when you apply the products, and the other is just directly to try the product, and the things that you are apply, like the exosomes, are still in all its potency. Is totally different thing.
Okay. Huge. So the key questions to ask your vendor. This we'll go through them quickly here, but before you commit to a purchase, to using it on your patients, what exactly is in this product and how to categorize it so you get this POA? What source? Can you provide
Mm-hmm.
proper documentation?
Mm-hmm.
What isolation or purification methods did you use?
Yeah.
Can you provide a COA from a third-party lab, right? What peer-reviewed studies support the efficacy claims for this specific final product, right? Not before.
Mm-hmm.
What is the
Yeah.
regulatory status? And have you received any FDA communication? Huge. What quality control and sterility testing do you perform on each batch? How should this product be stored, handled, and administered for best result? I mean, I think it's important for providers to know how to handle this product, right?
Mm.
What adverse events have been have been reported, and how do you track them? Which goes back to the data integrity, right? And make sure that you're collecting all that data. And most importantly, do you follow, not most importantly, everything is important here, but do you
Yes.
follow MISEV guidelines for characterizations and nomenclature?
It's fundamental. Yes.
And that website is right here at the bottom. As you guys can see, it's in the checklist. And a note on due diligence essentially is, as
Mm-hmm.
you guys heard, the exosome field holds an enormous promise for regenerative medicine in aesthetics, longevity, interventions. But it depends on us for scientific rigor, honest
Thanks.
labeling from companies, and responsible innovations, right? So this checklist will form as a very basic start, but it's your judgment, your clinical judgment, and ask questions that lead to better decisions.
Yes, that is fundamental.
If the vendor cannot clearly
Right.
and transparently answer these questions, run away, right?
Like,
Consider that signal to look elsewhere,
yeah.
and the right vendor. I like this last sentence here. The right vendor will welcome scrutiny because rigorous science is their competitive advantage.
Mm-hmm.
100%. If they're in this field, in this industry, they understand and they know that it comes with scrutiny, right? They're going to be criticized, asked so many questions, and you need to be ready, right? If you're in it.
Yes, yes. What is true also comes to the surface. So
Yeah.
at some point,
Yeah.
yeah.
Yeah. Well, Dr. Mazzeo, we've gone about 3 hours and we still have
Wow.
3 hours.
Wow.
And
Still people. Still, I can
yes,
see you. Still
yes,
people.
yes, there's still people. There's still almost everyone that has been with us. So yeah. So
That's great.
yeah, I've been
That's
getting comments, messages, text messages.
yes. That is amazing. I will. Yeah, I can answer at some point.
Yep.
Yes, if I have the question, I don't know, some way that I can reach them
I will. Yes.
and answer, or they
Yep.
can reach me directly, and yeah.
What I will do here is I have the list of everyone again that registered for this and is watching live. I'm going to just know that I'm going to be sharing that list with Dr. Mazzeo, and Dr. Mazzeo is going to be sending you guys, a welcome email or something. Just introduce. If you guys have any, please reach out to her directly,
Yes.
or you can reach me and I can connect you guys via email, whatever that is. I will be sending you guys this checklist that Dr. Mazzeo created. I just made it look fancy and nice for her. That's all I did. Um, and Dr. Mazzeo, any final last words that you have before we say adios?
No, the thing is, well, I'm I'm happy to have this opportunity and to be with you and all all the people there in this Sunday. So thank you for being there and for you to provide me this space to talk about this important topic.
You're,
Yeah, that is great. And more. There is more. So yes,
yes,
that's
there is.
3 hours, but is
There
yes,
is way more. And
more to discuss.
imagine like people go to conferences and they try to cover what exosomes are in a 15-minute lecture.
Yeah. No.
It's impossible, right? And so for any of you
Well,
guys,
that serves if you go for yes, you have one specific question or one specific or want to understand one specific thing, so you go there to that
yeah,
talk and yes, and also well, if you ask that specific question to the person that can resolve that question, that is also what yes the exosomes are for, and also all the networking that you can do there, but
yeah,
to for a conference to know everything, and also you must go there with at least a basic knowledge, yes, of what,
true.
yeah,
It's true.
that
Um,
is.
yeah, Dr. Mazzeo is doing a lot on her own. Obviously, I'm also doing a lot. I will continue bringing on other experts in the field. Even have two conversations talk. I would love to have debates. Um,
That would be great, yeah.
that'll be great, right? Like two scientists, talking about it, and imagine
Woof.
how much. Imagine that. That would be great. So
You
if
are
you
complicated.
well, no, not, but it's good because that you ask very good questions. Like the only questions that I can ask you are very basic questions, right?
Yeah.
Um, and so at that level requires two experts or three experts or more, right?
Mm.
So please, continue, subscribe or follow us and Dr. Mazzeo. I'll be sharing all that information here.
Yes.
And we will also, I do want to take a mention that we will be doing more videos together as well. As you can see, we love talking, and I love talking.
We have lots, yes.
Yeah,
Lot to do, yes.
yeah.
A lot
So
to do.
yeah, well, you guys have a beautiful day, and I look forward to seeing you guys on the next one. Okay,
Yeah, thank
adios,
you. Thank you
adios,
for being there.
adios, adios, adios.
Bye.